Can any of you explain the article on CRISPR genome editing that we might need to present?
I read that article 3 times and still cannot understand what CRISPR is, how it works, or even how it is used. Oh, Lord help me.
I still don't know what the Crispies is.
It's pronounced CRISPR, not Crispies, and I just read the abstract.
Okay guys, this isn't good enough. As intimidating as it may seem, we're going to have to go to his office hours to have him explain the article to us. At least he'll know we're trying and we might have something to present just in case.
You are right, as long as Nisa doesn't talk. Crispies? Seriously? You cannot even say the name correctly.
No time to bicker, guys. Let's get this over with.
Come in. How can I help you guys?
Professor, we all read the article on CRISPR but found it difficult to comprehend. With our presentation might being tomorrow, we were hoping you could go over the article with us to make sure we have a sound understanding of what to explain, just in case we might need to present.
I see. All right, follow me and I'll explain. What if we could live in a world where genetic disorders would become obsolete? A world where food would be more nutritious, tasteful, and adaptable to a number of environments, or one where cancer would be a disease of the past, where human babies could be custom tailored to one's preference in terms of height, eye color, and even intelligence.
That would be cool.
Shh! Let him explain.
Well, yes, it would be cool, and it is, especially since this reality may be closer than you think thanks to advances in genome editing using CRISPR. CRISPR, or clustered regularly interspaced short pelagic repeats, is a genetic engineering tool that can be programmed to target specific coded sequences of DNA to either cut or edit them, altering the genome of an organism. This is made possible through the use of a CRISPR sequence of DNA, a segment of DNA that contains short repetitions of base sequences with unique spacers in between, along with the Cas protein, which is also an endonuclease. This CRISPR gene editing system was first discovered in archaean and prokaryotes, acting as a primitive yet effective immune system against invading viruses. Prokaryotes and archaean that are able to survive invading viruses incorporate short segments of remnant viral DNA as a spacer in between the repetitive prokaryotic DNA sequences. This is done in order to detect and destroy similar viral DNA in case of future re-encounters. When DNA matching the spacer sequence enters a cell, the spacer DNA sequence is transcribed into CRISPR RNA, which is used as a precise targeting guide to matching viral sequences. The CRISPR RNA then binds to a Cas protein, which is produced by the CRISPR system of the cell and forms a complex that searches the bacterial cell for DNA matching this RNA sequence. Once located, the Cas protein complex binds to the matching DNA sequence. The DNA double helix then unzips, allowing the CRISPR RNA to bind to it to make sure the sequence is a match. If so, the Cas protein then cuts the DNA sequence in two, rendering the gene ineffective and protecting the cell from the viral attack. Yay! Because the CRISPR-Cas complex leads to the disablement of genes, researchers have attempted to create refined versions of the CRISPR RNA to edit the human genome. With these systems, researchers can permanently modify genes in living cells and organisms, which makes it possible to correct mutations at precise locations in the human genome in order to treat genetic disorders. One promising implementation of CRISPR genome editing is in the field of medicine, specifically dealing with the current donor organ situation. Presently within the United States, 140,000 men, women, and children are waiting on the organ transplant list as of August 2017 due to organ shortages. Researchers believe CRISPR may provide a solution to this scarcity by potentially allowing the growth of an unlimited amount of human organs viable for transplantation. Through the combined use of CRISPR Cas9 and induced pluripotent stem cells, also known as reprogrammed adult cells, the genomes of embryonic pigs are being edited to generate human organs. Pigs have been chosen due to the similarities between the functions and size of their organs to those of humans. In addition, their rapid development, large number of offspring per cell, and relatively short gestation periods make them exceptional hosts. The CRISPR genome editing system is used to disable targeted genes that code for the development of certain pig organs. This is accomplished by using a customized CRISPR guide RNA to cut out complementary DNA sequences within the pig embryo that code for an organ development. Induced stem cells are then taken from humans and injected into the embryo, where they replace the deleted genes for organ development. This allows for the possibility of functional human organs to be created. However, achieving blastocyte complementation for the development of a functional human organ in the chimeric pig has proven troublesome, as only about 10% of the total embryos implanted survived. Only 1 out of 100,000 cells from these surviving embryos were human cells. As I was saying, like most scientific advancements, CRISPR is no stranger to ethical scrutiny and controversy. One such controversy is regarding the possibility of having an off-target edit from using CRISPR. Despite the accuracy of CRISPR Cas9 complex and its ability to bind to a specific sequence of DNA and change it, we must think of the ramifications that may occur if Cas9 deletes unintended parts of the DNA strand. It is a possibility that the Cas9 protein may inaccurately remove a gene, leading to unpredictable changes that can bring about new and varying mutations. Therefore, in our attempt to improve the human species, we may inadvertently bring rise to new or worse genetic disorders that can be passed down onto future generations. Things go wrong in nature all the time, creating varying diseases and unexpected genetic alterations that we cannot control, which is exactly why we're using CRISPR. Nevertheless, just like these alterations occur from mistakes in genetic sequences, who says that a mutation in the highly accurate Cas9 protein won't occur, and if so, we may simply create unwanted variations of the very genes we are trying to correct or eliminate. Another troubling issue that lies in the development and use of CRISPR is the extent in which it will be used to alter our genes. Will CRISPR be used out of the urgency to treat genetic disorders or for more selfish and idealistic reasons such as trait selectivity? With our ability to use Cas9 to locate and change specific sequences in DNA, we have the ability to genetically engineer humans to our individual liking. If each of us is to gain the ability to pick and choose the traits we want our children to have, or to completely design new traits to better the human species, we could change the entire human genome, possibly creating a newer, better, and more adaptable species. Now, although CRISPR gives us the chance to choose the ideal traits a human should have, how do we determine these ideal traits? We are clearly different, we think differently, we value different things, we have varying perceptions of perfection. Therefore, how can we decide which of these traits are better than others? And in deciding this, and in choosing the better traits, we inevitably begin to remove the others from the gene pool almost indefinitely. So we need to start thinking about the consequences of changing our species to what we think is best. Are the changes we are making necessary, or are we just playing God? So, any question, guys?
Wait, so what's CRISPR again?